All right, I started the recording and I think we can slowly start and let the last participant to enter the meeting. So today we are hosting Dr. Santiago Navas. So I will start by introducing Dr. Navas and his background, and then we will listen to his presentation for 45 minutes, and at the end we will have a question and answer part. So you're welcome to unmute yourself and ask the question that you want to Dr. Nava, and feel free also to turn on your camera to make it more interactive at the end. So Dr. Nava graduated in zoology from the National University of La Plata in Argentina, and he obtained his PhD in natural sciences in 2008 at the same university. He is currently a senior scientist in parasitology at the Instituto Nacional de Tecnología Agropecuaria and a scientist at the Consejo Nacional de Investigaciones Científicas y Técnicas. His research focuses on systematics, ecology, control of ticks, and epidemiology of tick-borne pathogens. So within this subject area, he has published more than 250 articles, 2 books, 6 book chapters, and he delivered more than 60 presentations at academic conferences in different countries of America, Europe, and Africa. In addition to his academic research, Dr. Nava provides support as a consultant and advisor to vets, cattle farmers, pharmaceutical companies, and government agencies. Dr. Nava also coordinates different projects on ticks with medical and veterinary importance, and is an active collaboration with scientists from more than 20 countries around the world. So with that, Dr. Navas, I will let you take over.
Okay. Well, thank you so much for the invitation. My talk will be about species delimitation, the tools that we use for species delimitation in ticks, taking as example 2 neotropical group of ticks and 1 cosmopolitan tick, as Rhipicephalus bophilus microplus. And the first step is to do a distinction between the general concepts of species and the operational concepts to delimited species. The general concept provide an ontology, a theoretical ontology, from which operational concepts may be applied. Operational concepts are in fact the testable concepts, the facts that we test when delimited species. So, for example, there are many species concepts, and sorry, there are many species examples of species concept. And here you can see some of the more common species concept, like typological concept, biological concept, the famous biological concept of Ernest Mayr, the evolutionary concept, the phylogenetic concept, ecological concept, and phenetic concept. But only to mention some examples. But there are these more integrative concept, and I like to mention the proposal of De Queiroz, of Kevin De Queiroz. It's in my opinion a modern concept that Kevin De Queiroz say that species are separately evolving metapopulation lineages, or they are a complementary concept of that a species as an entity of organism that maintains its entity from other organisms through time and over space. So what is the important thing here? That all the other the other concepts, the previous concept that biological, genetic, phylogenetic, phonetic, are treated as that previously were treated as necessary properties, now are reinterpreted as contingent properties, that the species as metapopulation lineages may or may not acquire during the course of the evolution. So species are lineages that exist in nature irrespective of our abilities to find or to diagnose them. So this is the important point of the Queiroz idea, that discovered and naming species that we place in the Linnean category are testable hypotheses. So the more the criteria that meet, the higher of the degree of the corroboration of the hypothesis. So the previous species concept now are used a different line of evidence that we integrate in an analysis. So morphological difference, reciprocal monophily, pre- and post-zygotic reproduction isolation, fixed character state, ecological divergence are not concept, but they are different line of evidence that we use to test the hypothesis about species and to delimit species. So, and now I want to present three different examples to show us how this line of evidence can be used to delimit species. This one, the Amblioma cagenense complex, is the more easy example. It's an example to put in the books because it's so easy to see as all these lines of evidence are met to delimit species group. Here, previously, the Amblioma cagenense complex was considered one species distributed from USA to Argentina and Brazil. And this species and this taxon was considered one species, the same species, Amblioma cagenense. But after the study of different lines of evidence, we now determine that in fact the Amblioma cagenense complex represent a species complex, that at least 6 different species. So look at first, this is the first line of evidence, the phylogenetic evidence. When we use genetic data, we found that different population of metapopulation of Amblioma cagenense from different regions, from South America, from Central America, and from North America, from USA and Mexico, they form 6 different clades. One from the Yungas from Argentina and Bolivia, and areas from Central and South of Brazil, and other clade from Colombia, another clade from La Amazonia, another clade from the south of USA, Central America, and Western South America, in Ecuador, for example. The other clade with a population from dry Chaco in Argentina, Bolivia, and Paraguay, and the 6th clade from Peru. So here we have genetic evidence and phylogenetic evidence of differentiation between populations. Here the same, here the same information, and now in this phylogenetic analysis, different mitochondrial and nuclear genes were used, and here this is a work of Cotes Perdomo, where the mitochondria was used to do an phylogenetic analysis, and the results are the same. When we use the mitochondrial genome, is the same. You can see that all the taxons are when we use mitochondrial genes or some nuclear marker like here. Now the next line of evidence is morphology. When we take the other population, the other 6 population indicated by the genetic analysis, we found an exact correlation with the morphology. The morphology, the morphological difference is in agreement with the genetic difference. Here's some example. For example, in the ornamentation of the scutum of the males, in the puntations, difference in the puntation of the male, only to show some examples. Here, in the shape of the genital aperture of the female, also in the seta of the dorsum of the females. The point here is that the morphology is in line with the genetic, and what here the morphometric is the same. With the morphometric, we can find the same differentiation between the previous 6 population. So with discrete morphological character and also with the morphometric characters. And now we test another line of evidence, that is the biological, considering the biological species concept of Ernest Mayr about the zygotic isolation. Here we take 2 different populations. Here, I'm sorry, only to show the population from dry Chaco and the taxon from Yungas. When we try to cross the population from Chaco and the population from Yungas, here you can see here that the fertility efficiency index, you can see that the crosses of these 2 groups of population are not viable. So we found here a strong post-zygotic isolation. And here we have another line of evidence of differentiation: genetic, morphologic, and now we have the biological difference because these 2 populations, the crosses of these 2 populations are not viable. And here another line, we test another line of evidence, that is the ecological difference. We can see here, forget the names of the species, but here we have 2 groups of evidence. Amblioma tonelliae is the population from Chaco, and Amblioma sculptum is the population from Yungas. We can see here that the ecological preferences are so different between these 2 taxons. and also the another taxon of the group that is Amblyomma cagenensis sensu stricto, but forget the names. So what we have here, we have genetic evidence, morphological evidence, biological evidence, and ecological evidence to test the hypothesis that the taxon previously known as Amblyomma cagenense in fact represent a complex of species. So to determine that the species the Amblyomma cagenense complex is in fact a species complex, we use different lines of evidence, and the evidence was very strong. In all the different lines of evidence were very strong to indicate that Amblyomma cagenense is represented by at least 6 different species. And now another example. This is Amblyomma parvum. With the analysis of individual mitochondrial genes with sequences of individual mitochondrial genes, I am speaking about 16S and 12S, we found that the population of Amblyomma parvum from Brazil, here in the yellow, in the market with yellow here, the population from Brazil were so different, very different with the population from Argentina and Paraguay. Here using the information of the mitochondrial genes. When here you can see what I am talking about.
Hey, I just saw your call.
Yeah, I forgot your accident.
Oh, okay. It's okay.
Okay.
Excuse
Hey,
me,
if you didn't get a
Lydia.
chance, though, maybe you can have Henry take a look at the literature around
Hey, excuse me,
like,
Lydia, you're
immune
not
response
muted.
and race ethnicity.
Okay.
Okay,
Okay.
sorry about that.
And
Go ahead.
here you can see that the strong difference in 16S sequences between the population of Amblyomma parvum from Brazil and the population from Argentina and Paraguay. But when we incorporate a nuclear marker, the ITH2 sequences of ITH2, we found the pattern is not so clear like in the mitochondrial sequences. Here we found B is Brazil and A is Argentina. We found that, and CA is Central America. We found that the pattern showed by the mitochondrial genes is not the same when we incorporate nuclear markers. We see that the Brazilian populations are not monophyletic. You see here Brazilian population and here Brazilian populations. These populations are not monophyletic. So there is a controversy between the information obtained from 16S of 12S sequences of mitochondrial genes with nuclear marker as ITH2. So, because here, to difference with the Amblyomma cagenense complex, in Amblyomma parvum, the information given by mitochondrial genes are not the same that the information given by nuclear markers. So when we try to do a morphological analysis, we found that there are not morphological difference between Brazilian and Argentina and Paraguayan populations. Here is an analysis or morphometric characters that you see with B and with 8, there are no difference between the populations. And here we try to crosses and intercrosses and back crosses of the Brazilian and Argentinian population. So the crosses between the Argentinian and Brazilian population were viable. There's no problem with the hybrids. They were viable. So with the descendant of the hybrids, we did the back crosses. Look at all the combinations that we did, and between hybrids, and between pure lines with hybrid lines, and most of them were viable. So there are a clear difference with mitochondrial genes, but not in morphology and not in biology. They're not postzygotic isolation here. So when we try to do an ecological analysis, modeling, so ecological preference of different populations from Leopardum, we found that all of them prefer this, have the same ecological preference. They are distributed in the same ecological area. So they are not ecological difference. There are genetic difference in mitochondrial genes, but not in morphology, not in the crosses, and not in the ecology. That is, and here, another example with the cosmopolitan species, with the cattle teeth Rhipicephalus microplus, that previously was now as distributed in America, in parts of Africa, and in Australia. But now we know that the population from Australia, in fact, belong to a different species, Rhipicephalus australis. But when we analyze the mitochondrial genes, we found the population from America and Africa are so different with the population from Australia and New Caledonia. This is with mitochondrial genes with 16S and 12S. When we use in this paper of Barger, the result with the mitochondrial genomes are the same. The population from Australia here were different with the population of America, here represented by Brazilian sequences of the mitochondrial genomes. So the result with individual mitochondrial genes and with mitochondrial genomes are the same. So then, in this study in this work of Labruna, they did the crosses between Australian, African, and South American populations. South American from Argentina, African from Mozambique, and Australian. And they found that the crosses between African and South American population were viable, but not when the population of Africa and South America were crossed with the population from Australia. So here, the biological information is in agreement with the genetic information. And the same happened with the morphology. Look at here the difference, for example, in the males, about spurs in the palps, the difference in the spurs in the palps, this ventral spurs in the pulp between the Australian and the South American population. Here the length and the number of the zeta on the scutum and also on the scutum. Here the same. Look at the size of the zeta. So here the situation with Bophilis microplus, with Rhipicephalus microplus is the same that with Ambloma cagenense. All of evidence, morphological, genetic, and the biological support this differential between these two different species. So now the population from Australia belong to a different species that is Rhipicephalus australis. But the other point is about the threshold. When we did the genetic threshold to say this is the same species, this is a different species. But it depends of the genus. It's not universal. And here, look at here. Look at Amblyoma, 2 species from the Amblyoma cagenense complex. Amblyoma sculptum and Amblyoma Tonelliae. Here, the morphological, there is a morphological difference, yes, between Amblyoma sculptum and Amblyoma Tonelliae. The reproductive isolation is complete. The difference between mitochondrial genes, here I indicated as example the difference between 16S sequences is more than 7%. And when we did the phylogenetic analysis, we found that there is reciprocal monophily. So when we compare nuclear markers, as for example here ITS2, we found again that there is reciprocal monophily, and the genetic divergence is more than 6% between Amblyoma sculptum and Amblyoma Tonelliae. And as you saw previously, there are different about ecological preference between the two species. So all of different line of evidence go to the same point. They are different. They are different species. But what happened with Rhipicephalus micropluus and Rhipicephalus australis? There morphological difference? Yes. You see that I show it that difference, the morphological difference between Rhipicephalus micropluus and Rhipicephalus australis. And the reproductive isolation again is complete. But here we see that difference in mitochondrial sequences, in 16S sequences, is not more than 5, 6, or 7%. It's just 1.3 and 1.6. Is the difference between mitochondrial sequences is no more than 1.3 or 1.6. But there are reciprocal monophily when we did the phylogenetic analysis, and there's not divergence in ITS2 sequences, because the ITS2 sequences of Rhipicephalus species are so similar. They're not different. But to say that Rhipicephalus micropluus and Rhipicephalus australis are different species, we use the morphological difference, and we use the reciprocal monophily, and of course we use as line of evidence the reproductive isolation that exists between Australian and South American population of Rhipicephalus micropluus. But with Amblyoma parvum, we found that there are not morphological, there's not morphological difference, there is not reproductive isolation, there's not difference in ecological preference. But if you see in mitochondrial genes, the difference are higher than 3%, 3.2 in this case, in this example. And there is reciprocal monophily, but not when we use ITS2 sequences, as I showed it previously. There is not reciprocal monophily when we use nuclear marker as ITs2. So if you compare Rhipicephalus australis and Microplus, they are different species. Yes, we have different line of evidence like reproductive isolation and phenotypic difference to say that they are different species. But in the case of Amblyoma parvum from Brazil and Argentina, the line of evidence are contradictory because we didn't find difference in morphology, in reproductive isolation, in reproductive behavior. We only found difference in mitochondrial genes. So about the threshold, if you take the difference in mitochondrial genes are bigger for Amblyoma parvum than for Rhipicephalus microplus. So, I mean that there is not a universal threshold about genetic difference. We need to test another complementary lines of evidence to test the hypothesis about species differentiation. So, for example, the different here you can see that the difference in mitochondrial genes, in mitochondrial sequences in between Amblyoma parvum and Rhipicephalus microplus and australis complex is evident that the information is not the same. Here the difference in ITs2. Amblyoma parvum from Argentina and Brazil, they are the same species because they from a biological point of view, they are the same species, but the ITs2, the difference between the ITs2 sequences is bigger in parvum than in Rhipicephalus microplus, and Rhipicephalus australis. So this is the important to use different line of evidence to test hypothesis about species difference, because if I only take genetic difference, I can say that Amblyoma parvum from Argentina and Brazil are different species, but not. They belong to the same species because they are, from an ecological and biological point of view, they are the same biological entity. And to finish, this is the important point, that the information that DNA sequences, that information that give DNA sequences are not the same for different genera. For different genera, each genus is particular in with respect to the information that genetics marker give us. So, and thank you so much for your attention.
Thank you, Dr. Nava. Does anyone have any question about the presentation? Don't be shy. All right, maybe I'm gonna go ahead and ask a general question. Hold on, let me see. I have a question in the chat from Ricardo Paredes Leon, who asked why the COX-1 is not commonly used to separate tick species. That's a really good question.
About the COX-1, the CO1?
Yeah.
Okay.
Why is it not
yes,
commonly used?
The CO1 here, I didn't show the information about CO1, but is similar to 16S. Usually the information is similar to 16S. in some point is more polymorphic CO1 like 16S, but at least in my experience, the information is similar of CO1 with those of 16S.
We have another question from Luisa Dominguez. How do we decide how much information is enough to classify a new species?
It's a very, very good question. The point is to get the more line of evidence we have, more strong is the test. More strong is the test. So it's hard to say how much evidence we need. So of course, if you only have a morphology of, if you only have mitochondrial information, chlorine is not enough. So, for example, with barcoding, barcoding should be taken carefully, because in barcoding usually people use mitochondrial genes, like for example CO1, and here in this table you can see that the information of mitochondrial sequences, some is controversial, in some cases can be controversial.
So we have another question from Ethan. So now people are being really active. From Ethan Senbil, sorry for mispronouncing your name. So he said, thank you so much, Dr. Nava, for this great webinar. Could you please address the same situation for the biggest conflict ever of ticks, Rhipocephalus sanguinus, and its lineage, and propose recent species names? What is your opinion? Rhipocephalus linnaei and Rhipocephalus rutilus?
Yes, my opinion is, I agree that Rhipicephalus linnaei is a different species from Rhipicephalus sanguinus sensu stricto, because Rhipicephalus linnaei represent the previous taxon naming in several papers as tropical lineage. In that case, I agree with the conclusion that the Rhipicephalus linnaei is a different species of Rhipicephalus sanguinus sensu stricto. Remember, keeping in mind that Rhipicephalus linnaei represent to the tropical lineage of Rhipicephalus sanguinus. That I agree, and in that case, in this case, you have also different line of evidence, because there are reproductive isolation between the tropical lineage represented by Rhipicephalus linnaei and the temperate lineage represented by Rhipicephalus sanguineus sensu stricto, and there are genetic difference and there are ecological difference about distribution. And so I agree with the differentiation between within the Rhipicephalus sanguinus complex, the differentiation between Rhipicephalus linnaei, previously known as Rhipicephalus tropical lineage, and Rhipicephalus sanguinus sensu stricto, and the other species from the Rhipicephalus sanguinus complex as Rhipicephalus turanicus, Rhipicephalus rossicus.
Okay. We have another question from Tim Ebert. So would one weight the importance of different lines of evidence? Differences in 16S are worth twice a morphological difference, that sort of thing.
Yes, sorry.
No, that's it. I'm not sure if you understood the question, but basically it's asking if yeah, some difference are more important than other, specifically genetic and morphological differences.
Sorry,
So-
can you repeat the
Yeah,
question?
the question, yeah. Basically, he's asking, for example, some people state 16S are worth twice a morphological difference. So is that that kind of difference? So would one weigh the importance of different lines of evidence in examples such as he gave?
Yeah, morphology is so important. It's because when you found a constant morphological difference, at least in ticks, I am talking about ticks, and the morphology is so important. It's so important, and usually the genetic evidence is aligned with the morphology. So in many cases, for example, in Exodes, in the Exodes ricinosus complex, when you found different in sequences, then when we see carefully the morphology, there are also difference in morphology. So morphology is so important. It's a very strong line of evidence in the case of hard ticks. Is so important.
All right, thank you for your answer. Okay, one more question from Ricardo Paredes Leon. In your experience, the population of Amblyomma mixtum from Mexico can represent more than one species? Question mark.
At least with the information that they have, not. Amblyomma mixtum is the same taxon from the south of USA to north of South America, to Ecuador, at least with the information that I have, including Mexico. But again, I am talking about the information that I have. If maybe you have a new evidence or new information, but with the information that I have, the population of Amblyomma mixtum from the south of USA to Central America and north of South America in Colombia and Ecuador are the same species, the same taxon. I am talking about Amblyomma mixtum.
Okay. I was wondering about the people say thank you for your answer. You didn't talk about the because ticks are a vector, and so how the delimitation of species is important for ticks because they are a vector in terms of maybe their virulence and they the way they can acquire the pathogen and transmit it. So how that's your research and species delimitation link with the disease transmission and all these things?
Very nice question. For example, in the case of Rhipicephalus sanguineus complex, there are evidence that they are different in the vectorial competence to transmit Ehrlichia canis between the tropical lineage of Rhipicephalus sanguineus sensu stricto and the tropical lineage that now is named as Rhipicephalus linnaeus, and the point that is in amblyoma, for example, in amblyoma cajanense complex for rickettsia, there's not different. In the case of rickettsia, they are not different, but in the case of the rhipicephalus sanguineus complex, yes, there are different in vectorial competence. So in some cases there are difference in vectorial competence, but not in another relation, in another pathogen vector associations. So it depends of the pathogen, and it depends of the tick vector species. But yes, in some cases there are difference, but not in another cases.
Okay. Thank you. Well, I think, do we have more questions from the audience, or? I think we had yeah, we had the same. How important do you think vector competence is as a character?
Yes, it's the same question.
Yeah, kind of, yeah. But okay. All right. Well, I think if we don't have any more questions, I'm gonna thank you again for your presentation. I would just want to remind the people that if you know some people who were interested in watching the talk but were not able to, the link will be available on our website, so the Archaeological Society of America, the Archaeological Society of America. So we have a online website, and we also have a channel, a YouTube channel. So we will make this video available so that some of more people can watch it. And stay tuned, because we have one more seminar this year, and it will be in 1 month, and it will be that time about mites. So for the people who prefer mites, stay tuned. Well, with that, thank you so much, Dr. Nava, for your nice presentation, and thank you all for your question and your feedback on this presentation. I wish you a good day, and see you later.
See you later. Thank you.
Bye.